Okay, well, thanks a lot. I'm going to be talking about genome-wide association mapping in general, and then what I hope to do is to walk you through a real basic R script on performing a GWAS analysis. So we're going to kind of be doing it the old-fashioned way, but my goal is to help you understand how a script would run to do a genome-wide association analysis, so that, for one thing, you can maybe design a more sophisticated script to do different analysis on your own data, as well as get a better understanding of maybe some of the programs that use a graphical user interface on iPlant, what perhaps they do underneath the surface. And then my audience for this is going to be somebody who isn't terribly familiar with GWAS, maybe has heard of GWAS, maybe has used GWAS results, but wouldn't know how to do a GWAS analysis themselves. They don't know anything about the linear models actually underlying that analysis. So if you know a lot about GWAS, you know a lot about programming in R and how to do that type of stuff, you might be somewhat bored with this lecture, but that's okay because we'll be talking about some more specifics tomorrow. Okay, the first thing I wanted to point out that is this is a very big subject. There are lots of methods and software packages, and at least we're originally timed for about 75 minutes, and so I'm going to give this a pretty brief overview. What do we do in GWAS? Basically, what we want to do is we want to take all the variation for continuous trait or maybe even a discrete trait in our species or maybe in our germplasm or our population, maybe within a land race, and we want to associate that variation with the underlying genes that are generating that variation to give us, for one thing, to identify the actual functional variants that are generating the variation, and then also to learn something about the genetic architecture underlying that variation. How many loci are there controlling the variation for my quantitative trait? What is the distribution of allele frequencies at those loci? Do they interact with each other? What is the distribution of effect sizes, and so forth? Are there lots and lots of small effect genes, or are there relatively few large effect genes underlying the variation for that trait? And there are a number of ways to get after that. And association mapping is just one of those ways, but association mapping has several advantages relative to these other methods. For one thing, which is nicely summarized in this figure, association mapping has a higher resolution. If you're able to identify an association and you have rapid LD decay, which we'll talk about, chances are that that marker is going to be fairly close to the actual causal variant that you're after. Okay. Another major advantage is that research time is much, much less than when you're trying to develop a new mapping population from scratch. High density markers and association mapping techniques allow us to screen germplasm that's already existing and make associations right away. And then, of course, the other advantage is that if we're using all the diversity within our species, we're looking at all the genetic variation or the genetic variants underlying that variation all at one time. If we make a mapping population by crossing two parents, we're only looking at two alleles at every single locus. But if we are looking at all the variation within our species, we're looking at all the alleles that may be segregating in that species all at one time. So just contrast again with conventional QTL mapping, where we're making a cross. We're looking at two alleles at every single locus. With linkage disequilibrium mapping, we're using our entire, all the lines we have available to us, the entire amount of genetic diversity that we can gather. So maybe examples of diversity panels that could be used for this analysis. One example is 71 elite European maize inbred lines, maybe a diversity panel of 288 maize inbred lines. Some Arabidopsis lines were used in this study by Atwell et al. In 2010. And maybe we want to take 219 or 15 dogs from across 80 domestic breeds and use that panel for an association analysis. Just some examples on the types of panels that are typically used in these types of studies. So all this depends on this little thing that we call linkage disequilibrium. And essentially what this is this a non-random association between alleles at a locus. And the most basic statistic for LD is this D statistic. It's just the difference between the frequency of the haplotype. How do I just, I'm good? Okay. So it's just the difference between the frequency of the haplotype, okay? Say the haplotype between the big A and big B allele, and the frequency, this is the frequency we actually observed in the population, minus the frequency of that haplotype we'd expect by chance, okay, which is just the product of the allele frequencies. How often do these two alleles come together just by chance alone. And what is the difference between those two? Now, this is the most basic statistic. It ranges anywhere from 0.25 to negative 0.25, but it only actually reaches those limits when the allele frequencies are at 0.5. Whenever the allele frequencies are less than 0.5, the most that D can be is going to be less than 0.25. So a very convenient thing to do is to standardize this D statistic into something we call the R-squared statistic. Okay, and this is what you see in all the GWAS studies, is this decay of R-squared over physical distance. And this is simply just a standardized value of D here, okay, by the allele frequencies at that locus. Okay, so again, LD decay, how rapidly alleles start to disassociate with one another at different loci across a certain physical space depends on the population that we have at hand. Okay? In a biparental population, let's say a recombinant inbred line population, LD decays quite slowly, actually, okay? And it decays in a very predictable manner, okay? Because the only force that is going on here in this population that causes LD is physical recombination. So you can predict the amount of LD in a biparental population simply by the distance between the loci in centimorgans, okay? On the other hand, if we have an association mapping panel, maybe a quite diverse association mapping panel, maybe some maize inbred lines that come from all different types of land races across the entire world, we have lots of forces causing LD in this population, things such as population structure, which we'll talk about, genetic drift and so forth, and selection. And also, these diverse inbred lines share a common ancestor in the distant past. So there's been lots and lots of opportunity for historical recombination throughout the ages. And as a consequence of that, the LD in a panel like this drops off extremely rapidly, maybe as soon as 500 base pairs. Okay? So if you want to use a panel like this to do association mapping you better have very, very dense genotyping to ensure that you have a marker within 500 base pairs of your causal polymorphism that you're after. Another example in barley. Barley is much, let's say, the effective population size of all the barley germplasm in North America is much, much smaller. It's much, much less diverse. Okay? So LD decay, at least in this barley from the upper Midwest, decays more slowly. Okay? We have fairly decent LD, and I define decent LD as somewhere around 0.2, extending out to about 8 centimorgans, okay, on this barley consensus map. So I guess the point of this is that you can't set a certain marker density and say we need this marker density to this number of markers, and it'll work across all different populations and all different situations. LD decays at different rates in different populations, and you need to match the marker density with that specific situation, usually dependent on the diversity of your panel that you're using for association mapping. Okay, so to motivate the model that we're going to develop, let's just start with a real simple toy example. Let's say we have 500 random individuals from population phenotype, and this population is phenotyped and genotyped, okay? And we're scoring these alleles like a classical quantitative geneticist as A1 and A2, okay? And let's just say that we're going to genotype things ourselves in the lab, and we're going to do one marker just to save time and costs. And we score that marker, and every individual that's homozygous for A1, we're going to call 0. Everybody who's heterozygous, we're going to call 1. And everybody who's homozygous A2, we're going to call a 2 there. Okay? So the 0, 1, and 2 basically represents the allelic dosage of the A2 allele. Okay? And then if we want to find an association, we basically relate our phenotypic values of our trait of interest with the allelic dosage of the A2 allele, and we fit the simple linear regression model here, where we have y is equal to intercept, the slope of the effect of the allelic, of the relationship between the phenotypic value and the allelic dosage, and w is just the allelic dosage, and of course the residual. And we test this. We just want to see if the slope is different than 0 or not, and if it is, we declare that this marker is having an effect on our trait, presumably because it's linked to the causal polymorphism, or it's not. Okay? So is it that simple? I'm not sure, but let's go through this R script and do this simple example here. So first, let's fit this linear model function. So this LM function in R just fits a basic linear model. Okay. All right, so the reason why we can't just use that naive linear model is because in a diversity panel, we're nearly always going to have some form of population structure. Okay? And population structure is going to cause spurious associations between markers and QTL, whether or not they're spurious, so they may or may not be linked to one another. And this might be okay for making predictions, but if we actually want to map the causal polymorphisms underlying the variation in our trait, this is no good. It will end up in no man's land and waste a lot of time and money. Okay, so we need to account for these things somehow in a linear model. And this is just a really simple example to give you a feeling for how population structure causes LD between loci that are not actually physically linked with one another. So let's say we have two populations, okay? A red population and a blue population. And in the blue population, all the alleles, at the loci A and B, the big A and the big B alleles are completely fixed. Okay? So the only haplotypes that are present in this population are the big A, big B haplotypes. Okay? We have another population, the red population, where at these two loci, the small A allele and the small B allele are completely fixed. Okay? So in this population, the only haplotype we see is a small A and small B haplotype. Okay? So if we ignore this population structure and treat these individuals in these two populations as if they exist in the same population, okay? And we do a calculation here on the LD between these two loci, we'll end up with a D value of 0.25. Okay? You can count them up, you can do the math yourself. Okay? We have a frequency of this haplotype of 0.5, and the frequency of the A and the B allele is 0.5. Okay, so we do the math and we get 0.25. I didn't have to invoke any sort of actual physical linkage to show this result at all. Okay, it's just purely the result of the allele frequencies being different between the two subpopulations. And if we treat them the same, you get these two loci in LD with one another. Okay. All right, so to solve this situation when performing genome-wide association analysis, we need to basically add this term to the model to account for this statistically. So in addition to our marker effect here, we add a subpopulation membership effect, okay? And I don't want to get too tied down with the notation and stuff like here right now, but if you look at this in matrix notation, you can represent that in this way, where you have an intercept, a matrix of subpopulation values. Actually, I think it might be better explained with the next slide here. Okay, so this is actually an example here drawn out with the matrices shown. Okay, so we add to this as a fixed effect, the population structure effect. And this population structure matrix here can take a variety of forms. One of the forms it can take is the output from this program called structure, which goes through and it tries to assign individuals to certain subpopulations. It could take the form of a simple indicator matrix where you indicate this individual belongs to this subpopulation based on my prior knowledge, and this individual belongs to that subpopulation based on some prior knowledge. It can take lots of different forms. Okay, but the important point is that it exists in this linear model, and then if we have three subpopulations, we're going to have three subpopulation effects there, and then we still have our marker effect, and then in this situation we have two markers, and so we have a column for each marker where a 0 indicates the presence of one allele, and a 1 indicates the presence of another allele. Okay? All right, so that's what I would call sort of a gross subpopulation effect. But in addition to that effect within a diversity panel, we also have what we sometimes call cryptic relatedness or family structure. Okay? Not everybody clearly belongs to one subpopulation or another subpopulation, but there are differences in relatedness between individuals in a diversity panel. And this also can cause or generate some spurious LD, and so we also need to account for this in a linear model. Okay, and this is a nice picture of how the different versions of this might exist. So we have population structure on the y-axis, where at the top we have a very highly structured population, and then on the bottom we have less structure, but some family relatedness, where we have differences in relatedness between individuals. And to account for this, we're going to add another term to the model. This time this term is going to be a little bit different. Before we add the subpopulation effect here, Q and V, it's just another fixed term. It's just like adding, other similar marker effect terms of the model. I mean, not exactly marker effects, but we're treating them statistically in the same way. In this situation, we're going to add this term to the model, this polygenic effect, but now we're going to treat it as a random effect. Now this is going to give us a mixed linear model, okay? And when I say we're going to treat it as a random effect, we're going to assume that it comes from some distribution, in this case a multivariate normal distribution, and this variable here is going to have a variance-covariance structure proportional to this kinship matrix, okay? And this can take a variety of forms, but a simple way to think about this is that this kinship matrix basically contains the number of similarities across all loci in the genome between two individuals in the population. Okay, so this is going to model the fine scale relationships among all pairs in your diversity panel. And you can calculate this. I'll go over how you calculate that in a minute here. All right, so now we have this. We went from a naive, simple linear model to a somewhat complex mixed linear model. And this is going to be much, much more challenging to solve, especially when you have a large number of individuals, than your simple linear model, okay? And you can implement this in ProcMixed, in SAS, or any other mixed models package. But it's to take the inverse of that covariance matrix, especially when it's very, very large, is very, very computationally demanding. And so lots of tricks have popped up in the GWAS literature on how to take these inverses more efficiently. And one of those, which I use frequently and which is very commonly used in the literature, is called EMMA, or Efficient Mixed Model Association. And basically, rather than take just a straight inverse of that whole covariance matrix, they take this fancy eigenvalue decomposition approach to more efficiently do that. So I don't want to get bogged down in the details of this, but I just wanted to point you to this reference because this is the software package, or the R package that we'll be using in this tutorial. Okay, so back to modeling population structure and kinship. Again, there are a variety of ways to do this. You can use the structure output from this program called Structure. There's a very, very similar program out there called ADMIXTURE. These are basically just they're Bayesian clustering approaches that try to assign individuals to different subpopulations probabilistically. And what they essentially try to do is they try to attain Hardy-Weinberg equilibrium within the subpopulations. So in addition to those, you can take principal component analysis. So here what you would want to do is you want to do a principal component analysis, and then you take the first few top PCs and you fit those as your fixed effects to try to account for subpopulation structure, okay? And then for the kinship matrix, we can do something similar like a marker similarity matrix, where basically the kinship matrix exists of all the number of times two individuals share an allele at a locus across the whole genome. Or something more complex like a genomic additive relationship matrix, which is basically going to be a scaled and standardized version of the similarity matrix. Or you can use, if you have deep knowledge about the pedigrees in your population, you could use a pedigree relationship matrix. Okay? But a lot of times, at least in plant populations, we don't have this pedigree knowledge, and we just simply use the marker data to calculate this kinship matrix because it's easy and it's just right there. All right, so to fit this mixed linear model in EMMA, we can go through the rest of this script here. Multiple testing. And I have some code in here to perform a multiple testing plot, which isn't validated at all. I wrote it fairly quickly, so if you want to use this for a publication, please go through it and understand the source of it and make sure that it's correct in this before you use it yourself. I'm not going to have time to go through the details, but I do want to point out that with GWAS, a major hurdle is the number of tests that we're performing. We're performing 36,000 tests, and so we have this massive multiple testing problem. Okay? And
traditionally, how do we deal with it? Not traditionally, I should say, simplistically. The simplest method to dealing with this could be the Bonferroni correction. So we take our desired experiment-wise error rate, say it's 0.05, and to apply a comparison-wise error rate to ensure that we meet that experiment-wise error rate, we divide that desired experiment-wise error rate by the number of tests, and that will give us our new threshold to apply on a comparison-wise basis. But here we have 36,000 tests, and so if we divide through by 36,000, we come up with a very, very, very conservative comparison-wise error rate, which is going to reduce our power to declare significant results when significant results really are really, when the null hypothesis is in fact false. So, but it happens that in GWAS, it's actually invalid to use the Bonferroni correction because the Bonferroni correction assumes that all of your tests are independent of one another. And by definition, since these tests, hopefully we have high enough marker density in our GWAS study that these tests are not independent. They covary with one another. Okay? So using a Bonferroni correction is overly conservative. An option in using for QTL linkage mapping is to do permutation testing, but it's generally not valid for GWAS because you're not able to maintain that family structure when you permute the data randomly, 1,000 or 10,000 times. And so generally that's not valid, although there are some new methods that are being developed to deal with this issue. And another thing we can do is apply this false discovery rate, which was developed by Benjamini and Hochberg in 1995. Basically what you do here is you calculate the expected proportion of your QTL that are actually false, and you just live with that false discovery rate. Okay, you say at this p-value, I know that 10% of my discoveries are actually false, but that's okay, I'm just going to live with it. There's this other analysis here, which I wanted to go through in more detail, and which is the equation here that I have in this R script, which will calculate this statistic for you. It's from this paper anyhow. And basically what this does is it takes an eigenvalue decomposition approach to determine how many effective tests you have if they were all independent of one another. And then you take that number and you use it as you would in your Bonferroni correction. And it happens in this data set, we have 36,000 SNPs, but if my calculations are correct, we only have 619 effective SNPs. Okay, so then we use that 619 and correct for it as we would with a normal Bonferroni correction. And again, there are multiple software packages out there to use, lots of different methods, lots of methods that are much faster than the traditional EMMA. For example, there's this method called EMMA-max. There's this method called FastLMM, which Anne is going to talk about. And there are other software packages such as Tassel and Plink, which you can learn all about from those links I provided right there.